Hello my dear lovely students, welcome on this amazing platform of Physics Wallah, where I, Ayushi Agarwal, your botany teacher, is going to teach you the next chapter of class 11, that is mineral nutrition. But before starting with this chapter, let me start today's class with a very simple and a very old thought, that time and tide waits for none. I hope you all believe in this thought that if once the time is gone, it is never going to return back. So, it's a very important duty of each and every one of us to start respecting time. Try to complete your job within a span of time. And if you start following this practice, then mark my words, 100% surely guaranteed you all will succeed in your life. So please start respecting time, because once the time is gone, it is gone. Right? Okay, so with this, let's start the chapter that is mineral nutrition. In this chapter, my dear students, we are going to study about different nutrients which are responsible for metabolic activities of our body, for the growth of the body. Now, nutrients can be defined as those chemicals, those supplements which you either take in from your food, if not food, then either in the form of tablets also available in the market, which are used to build our body, which are responsible for promoting growth and metabolic activities in our body. If I talk with respect to plants, then plants, they absorb the nutrients from the soil or they prepare by themselves. And for why do they do this job? So that their body can grow properly. Now, nutrients can be of two types. Inorganic nutrients, also known as minerals, and organic nutrients. Organic nutrients like, for example, carbohydrate, fats, proteins, all these are your organic nutrients. About organic nutrients, you study in the chapter biomolecule. But what about inorganic nutrients? What about minerals like nitrogen, phosphorus, potassium? These are also very, very important, which you either take in the form of food or you take directly in the form of tablets available in the market. So now in this chapter, we are going to study about inorganic nutrients, which are commonly called as minerals. Hence, the name of the chapter is mineral nutrition. Clear? So see this. What are nutrients? Nutrients are those chemicals which are required for the proper functioning of the body. Number one point. Second, they can be either organic or inorganic. Organic nutrients like so and so, and inorganic nutrients like all the mineral elements. Our chapter, as I told you, is based upon these set of inorganic elements. Chalo, let's start. So before we start with the inorganic nutrients, the minerals, first we have to study a very famous technique called hydroponics. In the year 1860, scientist Julius von Sack, he demonstrated a soil-free cultivation system. He cultivated plant in a nutrient medium which is soil-free. Normally what we know? We know that if I have to grow a crop, if I have to grow vegetation, then that should be done on piece of land, because all the nutrients which the plant will require is present in the soil. So if I'll keep my plant in the soil, then surely it is the roots of the plant are going to absorb them from the soil and help in the growth. But for the very first time, Julius von Sack demonstrated a soil-free cultivation in a nutrient medium called hydroponics. Hydro means water and ponics means to grow. Hydro means water and ponics means to grow. Right? So, what is this? In 1860, Julius von Sack, a prominent German botanist, demonstrated for the first time that plants could be also grown soil-free in a defined nutrient solution correct, from their seed up to their plantlet stage. And once they have grown to plantlet stage, then you can transfer them to the field if it is required to do so. So this technique of growing plant in a nutrient solution in the absence of soil is known as hydroponics. Clear? Now I'm going to demonstrate the experiment which was performed by Julius von Sack. Chalo, let's do. So for his experiment, first important requirement is to have a beaker. Why beaker? Because in this only we are going to prepare a defined nutrient solution. Add water into it. Right? Now once you add water, then you keep all the nutrients, whatever mineral nutrients actually you know is found in the soil in known quantities. Remember, we are going to prepare a defined nutrient solution. Defined nutrient solution means what? When we say defined means whatever you are adding in the solution, the quantity of that is known. So whatever minerals I am going to add, their quantity, that how much I am adding, sorry, quantity is known. Right? So whatever minerals, like for example, if I'm adding calcium, nitrogen, magnesium, iron, then all how much I'm adding calcium, maybe 2 gram, iron, maybe 1 gram. So I'm knowing the concentration, I'm knowing the amount of nutrient which I am adding. So such a nutrient solution is known as defined nutrient solution. So in this beaker, we are going to add all the nutrients available in the soil, all the minerals available in the soil in a defined quantity. Right? So that means you have now prepared a defined nutrient solution. Number one thing. Second, now you're going to cover this lid, cover the beaker with the help of a lid having 3 openings. How many openings? 3 openings. So this is the lid which is having 3 openings. Why 3 openings? From first opening, you are going to place an aerating pump, or you may also known as stirrer. Simply you can say stirrer. This stirrer is used to mix the nutrients properly in the solution and also to have a proper aeration. See, when you're going to stir the mixture, there will be proper circulation of air as well as the nutrients will also get properly mixed in the solution. So this job is done by the stirrer, which is added through one of the hole on the lid. Second hole is used to keep your plant, the seed which you will let grow up to a plantlet stage. These are the roots of the plant which are immersed in the water, and above is the shoot system. Correct? And from the third opening on the lid, you are going to place a funnel. What this funnel is going to do? See, you know, whenever your plant will grow, it is going to take up the water, it is going to take up the nutrient. So a time will come that the nutrients and water in the beaker will become less. With time, with passing time, the concentration will become less. So you may need to add more amount of nutrients and more amount of water from outside. Now you cannot open the lid because plant is already placed. So you need a funnel to add anything extra which is required. Correct? I hope all the setup is clear, the entire setup. Plus you have to keep this setup under sun. Right? And you also have to cover this entire setup with a black sheet. So cover your entire setup with a black sheet. Why with a black sheet? Because to avoid any contamination due to the growth of bacteria or fungi. How? Because see, sun is already there, water is there, nutrient is there, sort of warm and humid condition will develop. That will lead to the growth of fungi and other bacteria. But now if I cover this system with black sheet, then light will only be received by the plant. My beaker will not get heated up. It will be protected from the black sheet. So here inside the beaker, temperature will remain cool so that it will not let any fungi or any kind of contamination. This is the entire setup that was framed by Julius von Sack called hydroponics. And now whatever plant require for its growth, nutrients already there, water already there. For respiration, roots need oxygen. So with the help of stirrer, you are also giving proper circulation, proper movement of oxygen. So roots happy, they are getting water, oxygen and nutrient. And if roots are happy, then surely the shoot system is going to flourish. The leaves need light for preparing food, so light is also there, and now this plantlet will grow and will be fruitful. So now you have done cropping, or you have done vegetation by replacing soil from water. Clear? So this was demonstrated by Julius von Sack, and this simple technique is known as hydroponics. Now, what are the benefits of hydroponics? The benefits of hydroponics says, benefits. Number one benefit is that you are culturing the plant, you are growing your vegetation by replacing soil. Now, when there is no soil, that means with the help of hydroponics, you have solved all soil related problems. Now, what are soil related problems? Like for example, in some places, soil may be thin, infertile, acidic, and in such soil, crop cannot grow. Now, when soil is unhealthy, it is not supporting the growth of the plant, there you can use hydroponic technique, right? Second, when soil is not there, then there is no issue of soil related microbes, which sometimes may attack your plant and may destroy them. Third, if such soil related microbes are not there and they are not attacking the plant, that means there will be no such use of herbicides and fungicides and pesticides. So the cost of production will also somewhat get reduced because you are not using external fertilizers and chemical pesticides, insecticides to protect your plant from any kind of insect or pest, as everything is done under control, not in the soil, right? So what are soil related problems which you solve by using hydroponic technique? Number one is sometimes soil may be thin, infertile and acidic, which does not support vegetation. Now in such area you cannot use the soil for vegetation, so you can replace soil from hydroponics. Second, no soil related growth of microbes. Microbes will not attack crop, will not damage the crop. Hence, use of chemicals like pesticides and insecticides will be avoided. Thus, use of chemicals like pesticides and insecticides will be avoided. Clear? So these are the three soil related problems which get solved when you use the technique hydroponics. Apart from this, my dear student, hydroponic technique has also helped to understand that which elements are essential for plant and which are not. Now see, my dear students, the earth according to NCERT has more than or near about 105 elements. In chemistry, you learn something about 118. The number of the elements discovered so far is actually 118, maybe more than that. But in your NCERT, in this chapter, the number is mentioned 105. So we will study that number of elements discovered so far is 105. Right? Now, out of these elements, In the protoplasm of the plant cell, near about 60 elements have been discovered. So if naturally Earth is having 105 elements, then plant cells, they actually have near about more than 60 elements. Now, plant is having near about 60 elements. Are all those elements essential? Are all those elements going to affect the growth of the plant body? Or only some are going to play a direct role in the metabolism of the plant? So surely all those 60 elements which are found in the plant body are not important. Some may be important, some may not be important. So which elements are important, which are not, that also we can demonstrate and tell with the simple experiment of hydroponics. So hydroponics is not only helping you to get rid of soil related problems, but it is also helping you to determine that which element is essential for the plant growth and which is not. How will you do this? Now, you have a defined nutrient solution. You have added all those 60 elements present in the plant body in known quantity. Now, you can vary the concentration of all elements one by one. Suppose there is calcium. Now, calcium is suppose initially 2 gram in this solution and my plant is growing healthy, no issues. Now, if I want to ensure that whether calcium is required by my plant or not, then I am going to vary its concentration. First, I may remove the entire calcium. Now, that means my nutrient solution is devoid of calcium. And if it is, that means this plant is not going to receive calcium now. And if calcium is important for plant, then surely the growth of this plant will then be retarded in the absence of calcium. If it is essential, and if it would not have been essential, so either you remove, increase the concentration, whatever you do, plant will happily grow. There will be no effect seen. That means that element will be non-essential. Correct? So on this basis, by varying the concentration of elements in the nutrient solution, we can also determine that which element is required by the plant body and which is not. So hydroponic also helps to determine the essentiality of the element. Correct? By varying their concentration in their defined nutrient solution. 2 major benefits of hydroponics, I hope they are clear. Now since hydroponics is so very beneficial, hence commercially we have tried hydroponics to grow 3 major crops of our day-to-day life, I should say: lettuce, seedless cucumber and tomatoes. These are the 3 commercial crops of our day-to-day life which are being now grown by the use of hydroponics successfully. And which are those 3 crops? Tomatoes, seedless cucumber and lettuce. That means this technique is successful. Now if you go and look for there's so many hydroponic farms also being established, right? That actress, yeah, Shilpa Shetty Kundra, that actress Shilpa Shetty has her own hydroponic farm. She has her own hydroponic farm which is very beautifully decorated. I remember in one of the video, she even showed her complete hydroponic farm where she grow small herbaceous plants like spinach, lettuce. She grow them with the help of hydroponics at her own farm, at her own home. So now hydroponics is becoming like a modern technique to replace soil than water. Right? I hope you have understood hydroponics and what are the benefits. Moving ahead, I already introduced the term essentiality. Right? Now we are going to focus on the same terminology, essentiality and non-essentiality. Now, essentiality and non-essentiality of an element, can be easily demonstrated with the help of hydroponics. Correct? So with the help of hydroponics, we can differentiate that which elements are essential and which are not. For that, first two statements I have already told you that more than 60 elements out of 105 present on the earth, more than 60 elements are found inside the plant cells, in the protoplasm of the plant cells. Now in this more than 60, I am counting even those elements which are present in very less amount, in trace amount. So the quantity of element, if it is even 10 to the power minus 8 gram per ml, itni kam quantity में भी if any element is present in the plant body, that too it is detectable, right? So if I'm saying that out of 105 elements discovered so far, plants have more than 60 elements in the protoplasm of the cell. So in this more than 60, I have counted all those elements which are found majorly in the plant body and even minorly also. Minorly means even if the element is present in 10 to the power minus 8 gram per ml. Such a low quantity is even detectable. Any element present in this small quantity inside the plant is also counted. Clear? So now from more than 60 elements, some may be essential, as I told you, some may not be. So first of all, how will you decide that which element is essential and which is not? You know the technique. You know the technique that with the help of hydroponics, by showing the variation in the concentration of any element, you can determine the essentiality. But what are the grounds? What are the parameters on the basis of which you are going to judge your element? That means I am talking about the criteria of essentiality. Yes or no? So criteria of essentiality was established by Arnon and Stout. There were two people called Arnon and Stout who established, the name is not written, let me write. So Arnon and Stout are the two scientists who tried to explain the criteria for the essentiality, that how can you tell that which element is essential and which is not. So there are some about 3 to 4 points, 3 to 4 criteria on the basis of which you have to judge. The number 1 point says the element must be absolutely necessary for supporting the growth and reproduction. The element, if it has to become essential element, if it really has to become essential, then that element should be directly involved in the metabolism of the plant body, like growth and development. The role of that element should be in the growth and development of the plant body. First criteria. Second, now in the absence, this is the second point. In the absence of that element, plant should not be able to complete its life cycle. That means if that element is not provided to the plant, then plant should fail to complete its life cycle. First point, it should be directly involved in the metabolism. Second, if that element is not provided, then plant will not be able to complete its life cycle. Third, if that plant should be if that element is suppose not provided to the plant, then there should be some symptoms visible inside the plant body. Like I said, if calcium is reduced in the plant body, right? If I'm not providing calcium to the plant, then maybe growth is getting retarded, maybe the leaves are not able to grow properly, something or the other is happening. So such elements whose deficiency, if I say, the better term to say is deficiency. So if some elements, if they want to become essential, then if they are not given to the plant body, plant should show certain deficiency symptoms. And those deficiency symptoms should only be overcome when that element is only being provided. ऐसा ना हो that deficiency symptom is of calcium and even if you are providing magnesium then too plant is happy. That means calcium is not essential. Even magnesium is able to do the same role as that of calcium. Yes or no? So the 3 points which I have told you for criteria of essentiality. Number 1, element should be directly involved in the metabolism of the plant. Second, plant should not be able to complete its life cycle if that element is not provided. Third, if that element becomes deficient in the plant body, some hunger signs, some symptoms should be visible. Fourth, the same element, if you want to overcome those deficiency symptom, then the same element should be provided to the plant. That means elements should be irreplaceable. They should be irreplaceable. Right? Elements should be highly specific. If calcium is deficient, if the symptoms are coming due to calcium, then only calcium can overcome those symptoms, not magnesium or iron. So that means elements should be highly specific and irreplaceable. These are certain features or criteria on the basis of which we judged our 60 elements found in the plant body, and then we found that out of 60, 17 elements are essential. 4 elements are such which are neither non-essential nor could qualify the criteria of essentiality. Such elements are known as beneficial elements, and all remaining, अब जितने बचे 17 plus 4 minus 60, the remaining one are non-essential. Clear? So 17 out of 60 elements found in the plant body passed the criteria of essentiality and became essential. Remaining all became non-essential except 4, which are called as beneficial. Correct? Chalo, now move ahead. Let's talk about the essential elements. Our chapter is all about to understand the roles of these essential elements, these 17 essential elements. First of all, these 17 essential elements on the basis of their quantity of requirement can be of 2 types, macronutrients and micronutrients. So what I have said, that these elements, essential elements, can be broadly divided into 2 categories on the basis of their quantitative requirement, macro and micro. Now what do you mean by macro? The term macro itself means large. So those essential elements which are required in excess of what? In excess of 10 millimole per kg of dry matter. Please remember this line. Very important. This 10 millimole per kg is a parameter that decides that which element is macro and which is micro. If any element is, the quantity of any element is required in excess more than 10 millimole per kg, then that is considered as macro. And the element which is required in less amount, less than 10 millimole per kg of dry matter, then that is considered as micronutrient. These micronutrients are required in very less amount, in trace amount. Hence, micronutrients are also known as, they are also known as trace elements. Clear? Next point. Out of 17, 9 are macronutrients, which are carbon, hydrogen, oxygen, nitrogen, calcium, magnesium, phosphorus, and I hope one more is left. Carbon, hydrogen, oxygen, nitrogen, calcium, magnesium, phosphorus and sulfur. So these are the 9 in all. Potassium. 1, 2, 3, 4, 6, 7. Yeah, one more you have to add, that is potassium. 1, 2, 3, 4, 6, 7, 8, 9. Yes, done. So 9 are the macronutrients, and out of 17, 8 are micronutrients, which are iron, manganese, boron, cobalt, nickel, chlorine. How much? 1, 2, 3, 4, 5, 6. Zinc, and I think one more I am missing. You can add it. So these are I have written over here 7, but yeah, one I hope molybdenum. So these are total 8 trace elements or micro elements which are required by the plant body. Now, micronutrients and macronutrients, there is one more point of difference apart from their quantity of requirement. My dear students, micronutrients are very specific when it comes to their amount of requirement. There is specific critical concentration of every micro element. If you provide the element above than critical concentration, that element will become toxic to the plant. And if you provide that element below critical concentration, then that element will start showing deficiency symptom. So the requirement of micro element is very, very specific. If you give more than that, it will become toxic. If you give less than that, then it will become deficient, and deficiency symptoms will be appeared on the plant body. But this thing does not happen with macro. Macronutrients, because they are always required in so excess that usually they never become toxic. They may become deficient, but when it comes to toxicity, then the toxicity of macronutrients is very rare. Why? Because they're always required in high quantity. All the time you give them carbon, plant is more than happy. Carbon in the form of CO2, plant will take all the time because it is going to use that CO2 along with water to prepare glucose. More the glucose, more the biomass, more healthy is the plant body. So will ever carbon become excess in plant body? Will carbon anytime will show toxicity symptom in plant body? No, because these are macronutrients and they are always required in excess. Clear? Yes or no? So let's write this point. The point says micronutrients, they are always required in specific amount. Right? If given lesser than that, like if given lesser than that, then it becomes deficient. And if given more than that, then they become toxic. Clear? But if I talk about macronutrient, usually macronutrient do not become toxic. As they are required in excess. Clear? So these are the basic differences between macronutrient and micronutrient. More you will learn about macro and micronutrient, their roles, but before going to the roles of macro and micronutrient, essential elements can also be divided into 4 categories on the basis of the function being performed. So 17 elements, first you have divided them on the basis of their amount of requirement. Second is on the basis of their function. These essential elements can be divided broadly into 4 categories. The number 1 category is structural elements, the elements which are actually employed in making the plant body. See, whether it is plant body or human body, our main structure, our framework is made with the help of carbon, hydrogen, oxygen, nitrogen. Because why? Our body is made up of carbohydrate, lipid, fats and proteins. Proteins are building block, carbohydrate, fats are required for energy, and all these macromolecules, they are made up of mineral elements carbon, hydrogen, oxygen and nitrogen. So these are the 4 macronutrients which are responsible for maintaining the structural framework. So the first category of essential elements on the basis of function is structural elements. So where is number 1? Yeah, the elements which are structural in nature, the 4 ones are CHON. CHON. Correct? Second important role of essential elements is in phosphorylation, in energy related function. So there are 2 elements which play very important role in providing or in development of energy. They too are magnesium and second is phosphorus. Why magnesium? Because you know that magnesium is the central metal atom of chlorophyll, and chlorophyll is only responsible for trapping light energy, and then that light energy is converted into chemical form, that is ATP and NADPH2. So if magnesium would have not been there, then no chlorophyll, no light energy, no photosynthesis. Correct? So magnesium, because it is the central metal atom of chlorophyll, hence it is also involved in ATP formation. And phosphorus, how it is involved? ATP, what does it stands for? It stands for adenosine triphosphate. So chemically to make ATP, we require phosphorus. So the 2 elements which are energy related, they are magnesium in chlorophyll and phosphorus in ATP. Clear? Third category on the basis of function is maintaining osmotic balance. See, the body is made up of cells. Cells need to maintain their water balance, which is very, very important. Cell cannot have excess of water and cell cannot even have less amount of water. So they have to maintain their water balance, which is called as osmotic balance, and it is very, very essential work. Yes or no? So the 2 elements which are mainly required for maintaining the osmotic balance are potassium and chlorine. And one very important example where you can prove that these 2 elements are required for osmotic balance is opening and closing of stomata. You remember, to guard cells, they become turgid when potassium and chlorine, they enter inside, creates hypertonic condition, opening of stomata. But once during night, there's no entry of such elements, potassium and chlorine, guard cells, they become flaccid, thus closes the stomata. And see, opening and closing of stomata is such a necessary event, which is under controlled by these 2 elements, that is potassium and chlorine. So third essential element or third important role of essential element is maintaining the osmotic balance. I think that is written on the next page. Okay. Fourth important function of essential elements is they help in activation of enzymes, promoters, catalysts. Like for example, I was talking about magnesium. Magnesium is not only related to energy, it is not only responsible for stabilizing the structure of chlorophyll, but it also helps in the activation of some very important enzyme like rubisco. Rubisco is the most abundant enzyme of the biosphere and it requires magnesium for its functioning. And you know, if rubisco doesn't function, there is no glucose formation. So magnesium is a very important element that requires for the stabilization of or the enhancing the activity of rubisco. So magnesium, apart from activating rubisco, it also activates another enzyme found in C4 plants called peptidase. See, magnesium is an activator for both rubisco and peptidase, that is phosphoenol pyruvate carboxylase. This enzyme is required in C4 plants for carbon fixation. So these 2 are very important enzymes which are activated by magnesium. Apart from this, there are so many Elements like nickel. Nickel activate urease enzyme. Zinc. Zinc, which is responsible for the synthesis of oxygen, also helps in activation of enzyme like alcohol dehydrogenase. Yes or no? Just see this. So both of which are critical enzymes in photosynthetic, that is written for magnesium. Now next, zinc is an activator of alcohol dehydrogenase. It also plays important role in the synthesis of oxygen. Molybdenum. Molybdenum. In this chapter, you will only see the role of molybdenum. It helps to activate enzyme nitrogenase. It makes nitrogenase enzyme. Hence, and nitrogenase, you know, it is very, very important for nitrogen metabolism. For converting atmospheric nitrogen into ammonia, nitrogenase is required. And to make nitrogenase, we require molybdenum. Clear? Yes or no? So these are certain elements. So one you can write, molybdenum responsible for activating nitrogenase. Right? So these are the list of elements which are required for proper functioning. So these are the categories basically of elements on the basis of their function. I hope you have understood this so far. Now moving ahead, osmotic balance, already told you. Moving ahead, now we are going to discuss about the roles of macro and micronutrient. Now you know there are 17 essential elements which are divided into two broad categories as essential and non-essential. And then essential again can be divided into two broad categories as micronutrients and macronutrients. Now you're going to see their roles. Everything, my dear students, I have taken directly from your NCERT. Nothing here and there. So whatever is written on the board, as you can see, is directly from your NCERT. And knowingly I have done this. Why? Because if you learn NCERT, if you learn the roles of nutrients from NCERT, then whatsoever question come from this section, you will be able to answer them. Right? So please students, learn whatever is given in this roles. Whatever is written on this page, you have to learn each and every line. Every year, questions are asked from this section only. Any of the line will be picked up and then question will be framed out of it. And if you have really learned it, you will be able to solve. And if you have ignored it, then your +4 marks are gone. Right? But we are good students. We will not ignore even 1 mark, which is important for NEET. Then that means we will study the roles in detail. Okay. So first is nitrogen. Nitrogen is a macronutrient which is required in excess. It is one of the structural element, yes or no, which is absorbed from the soil mainly as nitrate. Little bit of nitrogen can also be absorbed in the form of ammonium, but the major form of nitrogen which is absorbed from the soil is nitrate. Nitrate or nitrite. So mainly absorbed in the form of nitrate. Sometimes it can also be taken in the form of nitrite or ammonium. Clear? Now, moving ahead. This is the essential nutrient element which is required by the plant in greatest amount. Why greatest amount? Obviously, it is one of the structural element. It is absorbed mainly as nitrate ion. However, some can also absorb nitrogen in the form of nitrite or ammonium. Moving ahead. Nitrogen is required by all parts of the plant body, particularly the growing parts, the meristematic part, because their cells are dividing. When cells are dividing, then cells need to make new cell wall, cell membrane, and for this nitrogen is required. Clear? Nitrogen is one of the major constituent of proteins, nucleic acid, vitamins and hormones. So you have to remember this thing. Moving ahead towards the next role of nutrient, again a macronutrient, phosphorus. So similar to that of nitrogen, phosphorus is also a macronutrient which is required by the growing parts of the plant body, meristematic tissue. Phosphorus is a major constituent of cell membrane. You know, our plasma membrane, cell membrane is made up of phospholipid bilayer. So all cell membranes, they are associated with phosphorus. So phosphorus is a major constituent of cell membrane, certain proteins and all nucleic acid. Usually this all and certain terms are replaced. But remember, for protein, certain protein require phosphorus, but all nucleic acids are what? Nucleic acids, they are made up of three components: sugar, nitrogenous base and phosphate. So phosphate means phosphorus, and therefore all nucleic acids are made up of phosphorus, are having phosphorus in them. Clear? Now, phosphorus is absorbed from the soil majorly in the form of phosphate or hypophosphate. Clear? Moving ahead. Nucleotides, and it is required for all the phosphorylation reactions. You know, phosphorus is one of the energy related compound. Phosphorus is needed to synthesize ATP during phosphorylation. Correct? Moving ahead to the third important role of another macronutrient, that is potassium. Potassium, they are majorly absorbed in the form of K plus ions, and they are also required in abundant quantities because it is also one of the macronutrient majorly by the meristematic, that means growing tissues. They require potassium in huge quantity. Now what is the role of potassium? They help in maintaining the osmotic balance. They regulate the opening and closing of stomata. Anion-cation balance means osmotic balance, right? And they also help in maintaining the turgidity of the cell, opening and closing of stomata, and potassium may also be involved in some activation of the enzymes, right? Okay, moving ahead towards the next essential element, calcium. Calcium, which is mainly absorbed in the form of Ca2 plus ion from the soil. Second, it is also a macro element which is required in excess. Third, it is usually immobile element, hence it gets accumulated in the older leaves. It does not shift from older to the younger as and when required because they are immobile element. Fourth, calcium along with magnesium, they help to form the middle lamella. You know, middle lamella is made up of calcium, magnesium, pectate. Apart from this, this calcium also plays a role in the formation of mitotic spindles during the time of cell division. So therefore, calcium is also required by the meristematic tissues where cell division is taking place. Clear? Will you read it? See, calcium is absorbed in the form of Ca2 plus. It is required by the meristematic cells as it is required to synthesize cell wall. Cell wall, the cell wall synthesis may required hai. Middle lamella synthesis it is required. It gets accumulated in older leaves. It is required for the formation of spindle fibers, etc. I hope till calcium it is clear to everybody. Okay, so I hope calcium is clear. Now, moving ahead with the next important essential element, macro essential element, magnesium. My dear students, magnesium is also required in excess amount. It is one of the macro element which is absorbed from the soil in the form of magnesium ion, first thing. Second, you know, it is the activator of various important enzymes like rubisco and peptidase. Magnesium is also responsible for maintaining the structure of ribosome assembly during the process of translation. Two important functions. Next important function, magnesium also play a role during DNA replication. Apart from DNA replication, it is also required to maintain the middle lamella. As to make middle lamella, calcium along with magnesium is required. And it is also mainly required in the growing parts of the plant body, mainly meristematic tissues. Clear? So first, absorbed in the form of Mg2+, activation of enzyme, synthesis of DNA, RNA, constituent of chlorophyll, you know, it is one of the energy related compound, and it also maintains the ribosome structure. Moving ahead towards the next, sulfur, which is also an essential element absorbed from the soil in the form of sulfate ion, and it is majorly found in two amino acids, cysteine and methionine. Apart from making two essential amino acids, that is cysteine and methionine, the sulfur is also a main constituent of several coenzymes, vitamins like thiamine, biotin and coenzyme A, and it is also a component of ferredoxin. So you have to remember each and everything that is written on your board. Moving ahead with the next, iron. Iron, which is usually absorbed in the form of ferric iron. From the soil, it is absorbed in the form of ferric iron, and the major role of iron is first of all to activate some enzymes like catalase, right? So it is also involved in the activation of enzyme catalase that is required during your photorespiration process. Catalase is required. Apart from this, it is also essential for the formation of chlorophyll, right? And iron is mainly involved, like copper, wherever redox reactions are taking place. Wherever oxidation reduction is taking place, this iron and copper, these both elements, they play important role over there, right? Now, iron is one of the micro element, but even being a micro element, it is required in high amount. Matlab, it is like an intermediate between macro and micro. So that is why the first line of iron says that it is required in large amounts in comparison to other micronutrient. So it is kind of an intermediate between macro and micro. So iron is a micronutrient but required in larger amount as compared to other micronutrients. Moving ahead with the next, which is manganese. Manganese is absorbed from the soil in the form of manganese ion, and its major role is to help in making oxygen evolving complex that helps in photolysis of water. So the major role of manganese, which you have to remember, is splitting of water. It makes the oxygen evolving complex that helps in the splitting of water associated with photosystem 2 during photosynthesis. So along with manganese, even the same function is done by chlorine also. Apart from this, manganese also activates the enzymes required for respiration and nitrogen metabolism. Okay, moving ahead with the next, zinc. I already told you about zinc. Zinc, it is absorbed in the form of zinc ion, and it plays important role in activating enzymes like alcohol dehydrogenase as well as carboxylase, and it also allows the synthesis of auxin. Right? Copper. Copper is absorbed mainly in the form of cupric ion, and like that of iron, it is also involved in redox reaction. Wherever oxidation reduction has to take place, copper plays important role over there. Moving ahead, boron. It is absorbed in the form of borate ion. It is required for the uptake and utilization of calcium. So can you see, one element is dependent upon another element. Calcium uptake and utilization is dependent upon boron. Apart from the uptake and utilization of calcium, boron also help in membrane functioning, pollen germination, cell elongation, cell differentiation, and carbohydrate translocation. Clear? Yes or no? Moving ahead, molybdenum, absorbed in the form of molybdate ion. And you know, along with iron, molybdenum helps in making nitrogenase enzyme, which is required for nitrogen metabolism. So this is the important function of molybdenum which you have to require, which you need to learn. And even in this chapter, when I'll teach you nitrogen metabolism, there also the role of molybdenum will be recognized, will be recalled. Right? Moving ahead, the next important element is chlorine. Chlorine, which is absorbed in the form of chloride ions, Cl minus ions, and they are one of the micronutrient trace element which are required in specific quantity. They help in splitting of water. They also help in maintaining the osmotic balance. Along with potassium, they help in maintaining osmotic balance. They regulate the opening and closing of stomata, and they also make oxygen evolving complex, same as that of manganese, and helps in photolysis or splitting of water. Yes or no? I hope all the roles of macro and micronutrient are clear, and I expect you all that you will remember and learn all these things. Little information which is given in roles of macro and micronutrient has to be done by you. It becomes, even if you found it little, but when it comes from the point of view of NEET and other exams, then they carry huge weightage, I should say. Even if one question gets framed from these lines, then it is going to cost you +4 marks. Whether +4, -4, all it depends upon you. So please pay attention. This chapter, I tell you, mineral nutrition, is purely theoretical. The only little bit conceptual portion comes when we do nitrogen metabolism in nitrogen cycle. Otherwise, the whole section which we have done so far is purely theoretical. You have to remember that what are the total elements, what is the minimum concentration, criteria, etc., etc. Everything has to be learnt by you. So this chapter is theoretical, and hence it is more scoring. If you give little bit attention to this chapter, then whatever questions are framed from this chapter, you'll surely be able to answer them. Correct? Okay, moving ahead. Now, I already introduced you two terminologies, that is one deficiency and another that is toxicity. Now, what is deficiency? Deficiency is that condition in the plant body when required essential elements are not given in appropriate quantity. So there is usually a critical concentration of every element. Every essential element has its own critical concentration, that is the minimum concentration which is required by the plant body for its growth. And if you provide that specific element below its critical concentration, then surely the plant is going to show some symptoms called hunger signs, because the plant is not able to fulfill, or it's not able to fulfill the demand of that element. And such condition is known as deficiency, and those hunger signs are known as deficiency symptoms. Correct? Chalo, read it. The concentration of essential element below which the growth is retarded is known as critical concentration. The minimum value, the minimum amount of any essential element which you should provide to the plant, below which the growth gets retarded, plant shows hunger symptoms, are known as critical, or is known as critical concentration. Okay, next, the kind of deficiency. Now the most prominent and the most common deficiency symptoms are like chlorosis, necrosis, inhibition of cell division, delay in flowering, etc. The most prominent ones are like chlorosis, as I told you. Chlorosis means loss of chlorophyll. Now when the leaves will lose their chlorophyll, what will happen? The color of the leaf will change from green to yellow. The leaves are green in color due to the presence of chlorophyll only. Now when there is no chlorophyll, the color of the leaf changes from green to yellow, and slowly and steadily, when there is no chlorophyll, plant will not be able to perform photosynthesis, and it is going to later on damage the whole plant body. Clear? So what is chlorosis? It is the loss of chlorophyll leading to yellowing of the leaf. It is usually caused when some elements like nitrogen, potassium, magnesium, sulfur, iron, manganese, zinc and molybdenum, they get deficient, right? Next is necrosis. Necrosis is death of tissues. Death of living tissues. Now chlorophyll is not dying. Whole mesophyll cell is getting dying. Whole mesophyll cell is getting killed or is dying. That means the tissue is getting killed or is dying due to the lack of certain nutrients. In that case, the leaf color will change from green to brown. So what is necrosis? Necrosis is simply death of the tissue because of the deficiency of some elements like calcium, magnesium, copper and potassium. If this is happening in case of leaf, then the color of the leaf will change from green to brown. Correct? Moving ahead. Next important deficiency symptom is inhibition of cell division. Beta, cell division is highly energetically expensive and nutrient requiring process. Now, if there are no nutrients in the plant body, then surely plant cannot initiate division. And if there is no cell division, then automatically growth is getting retarded. Correct? So, next point, lack or low levels of NKSMO causes inhibition of cell division. Right? And one more symptom which is commonly observed is delay in flowering. Delay in flowering means reproductive phase get delayed. Flowering means reproduction. Now reproduction for formation of flower, fruit, etc. is also energetically expensive and nutrient requiring process. When there are no nutrient, when there are no proper energy, plant will not show flowering. And when there is delay in flowering, that means the vegetative phase is going to elongate and it will be of no economical benefit to humans. So delay in flowering is also one of the symptoms which is caused due to the deficiency of nitrogen, sulfur and molybdenum, that is NSMO. Correct? So these are the list of deficiency symptoms along with the elements responsible for this, as well as what do you mean by deficiency symptom and when it arises. This page, if you remember, all questions you will be able to solve. Correct? Okay, now moving ahead with the next topic, which is toxicity. Deficiency you have learnt. Deficiency means when the required concentration se kam aap element de rahe ho. Now what is toxicity? Usually toxicity is only of micro essential element. Macro essential element, they do not become toxic because they are always required in excess. Now, if, as I told you, there is a specific requirement of every trace element. If you provide them below it, it will lead to deficiency. But if you provide above it, then it will cause toxicity in the plant body. Likewise, in deficiency, you saw the deficiency symptoms. Similarly, when it comes to toxicity, then there are some toxicity symptoms also similar to deficiency symptom. Now, there is one specific definition to define toxicity condition in the plant. When the overall dry weight of the plant body get reduced by 10%, then that means the plant is showing toxicity of some or the other element. What is toxicity? When any mineral ion concentration in tissue reduces the dry weight of the plant by 10% is considered as toxic. If suppose there is an element called manganese, if it is becoming toxic, that means it is going to overall reduce the plant weight by 10%. Toxic ho raha hai, ma'am. The concentration of that element is rising. So why the dry weight is decreasing? Because one element toxicity is associated with many other elements deficiency. So one element toxic, other elements becoming deficient. So overall, the dry weight of the plant body gets reduced. Like for example, manganese. Suppose manganese is given above critical concentration and it is becoming toxic in plant body. So its own symptom is, if manganese become toxic, then it show symptom which is known as brown spots on the leaf. Suppose if manganese is becoming toxic and there is a leaf, right? So on the leaf, some brown spots will start appearing. Brown spots means what? Death of tissue, necrosis. Some brown spots will be observed which are surrounded by chlorotic veins. These are veins and veinlet network. And the veins will also start dying. So veins will also start dying. So you can say that manganese toxicity will be characterized by brown spots surrounded by interchlorotic veins. The veins which are usually green, they will lose their chlorophyll and they will become yellow. Like, wait, let me show you properly. If this is a leaf, proper leaf, healthy leaf, green leaf, the veins are also green in color, veins and veinlets, they are rich in chlorophyll. Now manganese toxicity is occurring. So what will happen? Some regions of the leaf, the death of tissue will begin, causing brown spots, appearance of brown spots. And wherever brown spots will appear, there the veins will also lose chlorophyll. And they will start turning yellow in color. So brown spots surrounded by chlorotic veins. Clear? Yes or no? Okay. Now this is the basic toxicity symptom of manganese. But manganese toxicity causes deficiency of three important elements, that is magnesium, iron and calcium. So one element is becoming toxic but three elements are becoming deficient. So overall the plant weight, the dry weight of the plant body is going to get reduced. Now what manganese does, my dear students, it inhibits the uptake of magnesium and iron. It inhibits their uptake from the soil. Now when plant is not able to absorb these two elements, then surely they are going to become deficient. One thing. Second, in case of magnesium, not only it inhibits their uptake, but it also does not allow the magnesium which is present inside the plant body to function. If manganese will become toxic, then binding of magnesium with its respective enzyme rubisco and peptase will also get inhibited. So manganese will inhibit the uptake of magnesium as well as will not allow binding of magnesium with its enzymes. Right? Third, how calcium becomes deficient when there is manganese toxicity. Beta calcium के absorption को नहीं रोकता manganese, but it stops its translocation. Now if suppose root cells have absorbed calcium, but it is not able to translocate it to stem and leaf, then what is the point of that absorption? Leaf and stem will show the deficiency symptom of calcium because they are not getting it. So like this, it shows deficiency symptom of calcium also. So if calcium becomes deficient due to the excess of manganese, then in that case its uptake is not getting inhibited, rather it inhibits the translocation of calcium. Translocation of calcium. Clear? So these are three elements which becomes deficient in the presence of the toxicity of manganese, and this is how the overall plant weight gets reduced by 10%. This you have to remember. This example is very, very important and you have to remember this example. I hope the concept of essentiality, difference between essential elements like macro and micro is also clear. The roles of various essential elements, then category of essential elements on the basis of function, what is deficiency and what is toxicity. All these things are absolutely, yes, even hydroponics. If it is clear, then now we will move ahead with the next important topic, that is mechanism of absorption of elements. Right. Now, what is mechanism of absorption of elements? In last chapter, if I talk about transport, then in transport, my dear students, you have already learnt about that minerals are usually absorbed actively due to root pressure. That means if minerals have to be absorbed by the root hair cells, then they have to spend energy, because the absorption is usually done against the concentration gradient, and these minerals, they usually then they move inside the plant body due to positive hydrostatic pressure, and the movement is symplastic. So absorption is also active and movement is also active. Correct? Now more we are going to learn about the same topic, the same concept in this chapter as well, under the heading mechanism of absorption of elements. Right? Which you can see I have divided into two phases, initial phase and metabolic phase. So to understand the initial and metabolic phase, chalo, let's try to first draw the diagram. Here only I'll draw the diagram and then I'll write it. Suppose this is my epidermal cell, which is associated with cortical cell and then pericycle cell, so on. Limited only I'm drawing, I'm not drawing the entire anatomy. You might be able to recall this diagram which we did while I was explaining you apoplastic and symplastic movement in transport chapter. Right? Now, the epidermal cells, they are associated with their unicellular outgrowths. Correct? So these are the root hair. Each living cell is connected through plasmodesmata. So these are plasmodesmatas, the openings. Right? This is the dead region. This is the cell wall. And this is the plasma membrane. And just you can say it's the space between the cell wall and all intercellular spaces. All this, these are intercellular spaces. Now, root hair, when they initially absorb the nutrient from the soil actively. Root hair are absorbing nutrients along with dissolved water actively against the concentration gradient. So first way, where are they going to pump those nutrients? In the cell wall, in the intercellular spaces, in the dead regions of the cell, in short. So that first stage of the transportation of ions, once they get absorbed, is the initial phase. So in initial phase, the ions are first pumped into the dead regions of the cell, that is cell wall and intercellular spaces. Here the ions will come along with the dissolved water. Now, once the initial phase is done, this is the initial phase where the ions, they are pumped or they are pushed inside the dead regions of the cell. Correct? This movement will be surely apoplastic, passive, right? But once, now where these ions, will they just go to the xylem cell through the apoplastic region only, or will they enter inside the cell? So yes, all these ions will now enter inside the cell. That means they change their phase from initial passive phase to metabolic active phase. Why? Because now the ions are entering inside the cell, in the living space of the cell, right? Where the movement will change from apoplastic to symplastic. The movement will become comparatively slower, as resistance will be offered to the movement of water along with mineral. Now, each cell is going to take up the required minerals and will pass then to the another cell through plasmodesmata. So first phase for the movement of ion inside the plant body is apoplastic, which is initial phase, which is passive, which is comparatively faster, and then followed comes your metabolic phase, where the movement becomes comparatively slow, energy is required, and the only reason is that the movement is becoming symplastic. Clear? So what are the two phases for the movement of water along with dissolved mineral in the plant body once it is absorbed through the root hair? So there are two phases, initial phase and metabolic phase. Initial phase is characterized by the movement of ions and water in the dead regions. First thing. Second, the movement will be apoplastic. Third, passive. Fourth, faster. Then water will along with the minerals will enter inside the cell, where the movement will become comparatively slower because the mode changes from apoplastic to symplastic. The movement will become slower. It will become active, and here the movement of ion from one cell to another, this movement is known as flux. Influx, efflux. Influx means if ion is coming inside, so this is influx. Now ion is going outside from the cell, then it is called as efflux, right? So in that case, in case of metabolic phase, the movement of ion along with dissolved water is known as flux, influx and efflux. Clear? Yes or no? And what will you write for initial phase? First of all, it is apoplastic. Why? Because the water is moving along with minerals through the dead regions initially, and then it becomes symplastic. Hence it is fast as well as passive. Right? So these are the two phases for the movement of ions along with dissolved water. With this, your mechanism of absorption of element is also done. Now the next very important topic, which is purely conceptual and you have to understand, is metabolism of nitrogen. This is the very important section of this chapter where you actually have to focus. So students, please focus on this topic as well, that is nitrogen cycle. Now we are going to start with the main part of the chapter that is nitrogen metabolism, where you are going to study that how atmospheric nitrogen is converted into usable form of nitrogen compounds which are used by the plant body in order to produce proteins, and once those plant bodies and animals they die, then again those compounds of nitrogen they are decomposed to release the nitrogen back in the atmosphere. All these things, the process of nitrogen metabolism, the utilization of nitrogen, that we are going to study now in this chapter, which is very important, easy, purely conceptual, and if you understand it once, then it becomes very easy to learn as well and to remember as well. So I hope you all are ready. So before starting with the nitrogen metabolism, there are 3 to 4 points which you should know related to nitrogen. So first of all, nitrogen, it is a divalent molecule. The two nitrogen atoms, they join together with the help of 3 covalent bonds. As a result, the dissociation energy of nitrogen molecule is quite high. You have to break, if you want to separate both the atoms of the nitrogen, then you have to provide high amount of energy in order to break all those 3 covalent bonds. Now in the atmosphere, nitrogen is about 70%, not even 70%, if I make sure that it is about 78%. So atmosphere has 78% of nitrogen, which is very huge. But still, nitrogen is one of the limiting element present in the plant body. It is one of the limiting element which is known. The other two limiting elements are phosphorus and potassium. Now, when nitrogen is so excess, it is about 78% in the atmosphere, then why it is still considered as limiting? Because the nitrogen which is available in the atmosphere is not present in its usable form. Right? The atmospheric nitrogen, the gaseous form of nitrogen, cannot be used for protein synthesis. First of all, that atmospheric nitrogen should be converted into usable form of nitrogen, compounds of nitrogen like nitrate, nitrite or ammonia. Then only plant can take it and utilize it for protein synthesis. Otherwise, the atmospheric nitrogen, be it 78%, 70, 85, whatever percentage, but if it is not present in its compound form, then it is of no use. So first reason that why nitrogen is a limiting element even when being excess in the atmosphere. Now my dear students, in the soil, already the compounds of nitrogen are less, they are limited. Now in the soil, roots not only have to, there is not only one challenge for the roots of the plant, that is nitrogen is limited. Correct? But the second challenge is that the roots also have to compete for that limited nitrogen compounds with other soil microbes. There are so many soil microbes living in the soil who also try to absorb those nitrogen compounds for their purposes. Now roots also want those nitrogen compounds, other soil microbes also want that nitrogen compound. So already the nitrogen compounds are less in the soil, plus roots have to compete with the other soil microbes for that limiting nitrogen compound. So this becomes the second reason that why nitrogen, even being in excess, still roots have to compete for that limited nitrogen. Clear? So what have I told you so far? First of all, nitrogen exists in divalent state. Nitrogen molecule is made up of two nitrogen atoms that are joined with the help of three covalent bonds. Therefore, the dissociation energy of this nitrogen molecule is quite high. Second, in the atmosphere it is about 78%, but still it is, after even having high concentration of nitrogen in the atmosphere, but still it is limiting. Why is it limiting? There are two reasons. First, the available form, the usable form, the form of nitrogen which should be used by the plant is not present in the soil. So the nitrogen is usually not present in usable form, right? It's not present in usable form. First point. Second, the roots of the plant have to compete. Root of the plants have to compete for the limited nitrogen in the soil. Compete with the microorganisms, with the soil microbes for the limited nitrogen. Nitrogen compounds in the soil. Correct? Yes or no? Now this is the few points which you should know regarding nitrogen. Now we are going to understand the nitrogen metabolism, but for that you should know the nitrogen cycle, that how nitrogen metabolism has to take place in the form of cycle, right? Chalo, so here I'll be drawing the nitrogen cycle for you. Now my dear students, nitrogen cycle is completed in three pools. Pools here does not refer to swimming pool. Pool means like, for example, if I say atmospheric pool, so the area which is rich in gases, that is atmospheric pool. Then second is soil pool. Soil pools means area which is rich in soil and soil related microbes and nutrients. And then the third pool, which in nitrogen cycle we are going to talk about, is the living pool. Living pool includes all those organisms which have life, like plants and animals. So nitrogen cycle is actually completed in 3 pools. So the first one from where the cycle start is the atmospheric pool, because the major source of nitrogen is atmosphere. Atmosphere has 78% of the nitrogen, right? Now this atmospheric nitrogen from the atmospheric pool wants to come to the second pool, which is your soil pool. Now how will it enter into soil pool? By the method of, or by the process of fixation. The nitrogen, atmospheric nitrogen, will be converted into some usable form of nitrogen, some compound form of nitrogen, by the process of fixation. This fixation can be done by 3 ways: biological nitrogen fixation, industrial nitrogen fixation, and electrical nitrogen fixation. When I specifically talk about biological nitrogen fixation, then nitrogen is converted into ammonia. So if the question comes that what is nitrogen fixation? The nitrogen fixation you will define as conversion of atmospheric nitrogen into some usable compounds. But if specifically if the question mentions that what is biological nitrogen fixation, then in that case your definition will become conversion of atmospheric nitrogen into ammonia. Specify, you will, in that case you will specify that which compound of nitrogen is being formed. So in case of biological nitrogen fixation, the first stable product is ammonia. Clear? So now you are having atmospheric pool, nitrogen that is going to come in the soil in the form of ammonia, if that is done with the help of biological nitrogen fixation. I'll teach you. This biological nitrogen fixation is there in your syllabus. We'll do this concept in detail. Apart from this, there are 2 more ways of nitrogen fixation. One is industrial nitrogen fixation. You must have studied in chemistry Haber's process. So what happens in Haber's process? Nitrogen and hydrogen, they are joined in the ratio 1:3 to obtain 2 molecules of ammonia. That means for industrial nitrogen fixation as well, the first stable product is ammonia. Third is electrical nitrogen fixation. Electrical nitrogen fixation means when in the atmosphere there is lightning. When lightning occurs, students, the temperature in the atmosphere becomes very high, which is about 2000 or literally very high. I will not specify the exact temperature range, but yes, when lightning takes place, then the temperature in the atmosphere is too high. In that high temperature, the nitrogen gas and oxygen gas, they join together to form oxides of nitrogen, which then with rain will come down in the soil. So like this, electrical nitrogen fixation also takes place, but in that case, the first stable product is not ammonia, rather it is other oxides of nitrogen. Clear? Now you can see by any methods, by either biological, industrial or electrical, we have converted atmospheric nitrogen into compounds of nitrogen. Mainly I will be talking about ammonia. So if I talk about ammonia which is there in the soil pool, now soil has ammonia, but there is one trouble, and that trouble is that the roots of plants, they cannot absorb nitrogen in the form of ammonia. Even if ammonia is there in the soil, plant will not prefer this ammonia. Why? Because plants, they absorb nitrogen mainly in the form of nitrates. So next step in the cycle will be conversion of this ammonia into nitrate. Clear? So conversion of ammonia into nitrate is known as nitrification. And why this process is required? This process is required so that roots can easily absorb the nitrogen for their protein synthesis. So now ammonia will be first oxidized. to nitrite, and then further it will be oxidized to nitrate, right? And this process is known as nitrification. So first you performed fixation, then you performed nitrification, so that ammonia can be converted into nitrate. Right now, my dear students, I am only telling you the terminologies of the process that takes place during nitrogen cycle. Each step we will do in detail. But abhi ke liye for time being, just listen to the steps. Number one step is nitrogen fixation. Second step is nitrification. Now during nitrification you obtain nitrate. This nitrate in the soil or from the soil will be uptake by the plants. The root of the plants will absorb these nitrate, and now the nitrate will pass from the soil pool to living pool. So here comes the third pool of the nitrogen cycle, living pool. Now plants, they have absorbed the nitrate, the nitrogen form, that is nitrate. They will use there for protein synthesis. They will build their body. From the plant, that will be passed to animals. Right? Now a time will come, then these plants and animals, they will die, and then decomposition will take place. Decomposition means ammonification. Usually you must have observed when there's an animal die, or there is some decomposition or decaying going on, maybe on the roadside when you're passing, then you can smell a peculiar there's a peculiar smell when you're passing around and there's some decomposition or decay is going on. That peculiar smell, that bad smell, that odor is of ammonia, because when the dead organic matter is decaying, it releases ammonia. Correct? And this process is known as ammonification. So now ammonification will take place to release back ammonia when these organisms, when the living organisms will die. Right? Now, in the soil, my dear students, now to complete the cycle, atmospheric nitrogen should be produced. You have released ammonia, but where is atmospheric nitrogen? So, in the soil, there are some microbes like thiobacillus and pseudomonas who convert nitrate back into nitrogen. So, some amount of nitrate is absorbed by the roots, and some amount of nitrate is denitrified back to nitrogen, atmospheric nitrogen, with the help of some denitrifying bacteria like pseudomonas and thiobacillus. So these are the important steps. Number one step kya hai beta? Which is the number one step? Fixation. Second step, nitrification. Third step, uptake. Fourth step, transportation from plants to animals. Then fifth step, death and decaying of organic matter. Sixth step, that is your denitrification. So with these many steps, the nitrogen cycle is completed. So this is just the outline of the nitrogen cycle. Now one by one, we are going to see all these steps in detail. First, starting with nitrogen fixation, and even in nitrogen fixation, the important one which we have to discuss is biological nitrogen fixation. Okay, so are you all ready? Yes, very good. So now let's start with biological nitrogen fixation. Now, what do you mean by biological nitrogen fixation? When nitrogen fixation is done with the help of living organisms. Bio means living, and when some living organisms are doing this purpose or this conversion, then that process is known as biological nitrogen fixation. Now, all organisms are not capable of doing this process. There are only some bacteria, there are only some prokaryotes who can only perform biological nitrogen fixation. No eukaryote is capable of doing this process. Why? Because to perform this step, biological nitrogen fixation, there is some specific enzyme required called nitrogenase. And this nitrogenase enzyme is only found in bacteria. That too few bacteria. We do not have nitrogenase. So in my body, no nitrogen fixation can take place. From outside only, I have to take my protein. But there are some bacteria, there are some prokaryotes who have this special enzyme called nitrogenase, which help in the conversion of nitrogen into ammonia. Clear? So now you can say, how will you define biological nitrogen fixation? The process by which atmospheric nitrogen is converted into ammonia. So we can say that the first stable product of biological nitrogen fixation is ammonia. Right? First point. Second important point. Yeah, second important point is only some prokaryotes can perform this activity, are utilized for this purpose. Why? Because only some prokaryotes have the special enzyme called nitrogenase. So this enzyme is mainly required to perform this conversion from nitrogen to ammonia. Now I am going to tell you the list of those bacteria which are going to help in nitrogen fixation. This list you have to remember. Questions will only be asked from this list. Chalo. So now I am going to show you the list of bacteria which are being used. Now some bacteria, they may be aerobic or anaerobic. Some bacteria, they may be aerobic and they may fix nitrogen in free living state, or they may form symbiotic association with other organisms for nitrogen fixation. So everything I'll be telling you while I'll be mentioning the organisms in my list. First I'll start with Azotobacter. So Azotobacter is a bacteria, Azotobacter, along with another bacteria called Beijerinck. So Beijerinck and Azotobacter are two aerobic bacteria. They need oxygen for their respiration and they fix nitrogen in their free living state in the soil. These two organisms, they do not need the help of any other higher organism for their nitrogen fixation. They have their nitrogenase, they are free, they have their nitrogenase, and independently in the soil they can convert nitrogen into ammonia. Third example is Clostridium. Clostridium is a anaerobic free living nitrogen fixer. Clostridium. It is also free living nitrogen fixer, but itself it is an anaerobic bacteria. It respires in the absence of oxygen. So these three along with Rhodospirillum, Clostridium and Rhodospirillum. Rhodospirillum is also an anaerobic nitrogen fixer which fix nitrogen usually in free living state. Correct? Okay. Fourth now. Apart from these examples, there are some cyanobacteria also. These are eubacteria. Now there are some cyanobacteria also like Nostoc and Anabina who can also perform nitrogen fixation both in free living state and in symbiotic state. Right? So next is some cyanobacteria, blue-green algae like Nostoc and Anabina. They are aerobic organisms. They can also perform photosynthesis, oxygenic photosynthesis, and they can fix nitrogen both in free living as well as symbiotic, like in the roots of cycas. Normally Anabina and Nostoc, if they are free living in the soil, then too they can do the same function. But if they get associated with the roots of higher plants like cycas, then there also they will help in nitrogen fixation. So these are the list of bacteria and cyanobacteria which have nitrogenase enzyme and have the capacity of fixing nitrogen, either free living or symbiotic. Now two more examples I have in my list, Rhizobium and Frankia. Rhizobium is a rod shaped bacteria which usually is free living, lives normally in the soil, but when it has to perform nitrogen fixation, it gets associated with the roots of leguminous plants. Whereas Frankia is also usually free living, but when it has to perform nitrogen fixation, it gets associated with the roots of non-leguminous plants like Anlus and Casuarina. So two more names you have to remember, very, very important. Here I'll write Rhizobium and Frankia. Rhizobium gets associated with roots of leguminous plants, right? Like your pea, lentils, all the members of Leguminaceae family you have already studied. Now, Frankia gets associated with the roots of non-leguminous plants like Alnus and Casuarina, and then they both help in fixation of nitrogen into ammonia by the process of symbiosis. They form symbiotic association and they help in nitrogen fixation. Correct? Yes or no, everyone? Okay, so this is the fourth point. Next point in biological nitrogen fixation which you have to remember is some features about the enzyme nitrogenase. If I talk about this enzyme nitrogenase, bacho, so this nitrogenase enzyme is actually made up of two elements, that is molybdenum and iron. So it's a moiety of, it's a heterodimeric protein which is made up of two elements, molybdenum and iron. First point. Second point, this nitrogenase, my dear students, it is only active under anaerobic condition. If conditions around the cell which has to perform nitrogen fixation is aerobic, then this enzyme will become inactive and there will be no such process. So this is a very peculiar property shown by enzyme nitrogenase, that is, it is sensitive in the presence of oxygen. For functioning of nitrogenase, anaerobic conditions are must. So you can write that this enzyme is sensitive to oxygen. That means for functioning it requires anaerobic conditions. Anaerobic conditions should prevail in the cell which is about to perform nitrogen fixation. Clear to everybody? Okay, so this is something which you have to remember about the nitrogenase enzyme which is found all in these bacteria and that help in nitrogen fixation but in anaerobic condition. Now my dear student, this process of nitrogen fixation is an energetically expensive phenomena. It is energetically expensive phenomena. What do you mean when I say it is an energetically expensive phenomena? It means that to convert nitrogen into ammonia, lot amount of energy ATP has to be broken down. And I tell you, for one molecule of nitrogen to produce 2 molecules of ammonia, 16 ATP are required. So you can see it is such an energetically expensive phenomena in the presence of the enzyme nitrogenase. So I'll show you the reaction of biological nitrogen fixation and the role of energy. Chalo. So let's move ahead. So now there's equation of biological nitrogen fixation. Now one molecule of nitrogen will combine with 4 molecules of hydrogen. In industrial nitrogen fixation, the ratio of nitrogen and hydrogen combination is 1:3. But in natural condition, the combination to produce ammonia for nitrogen and hydrogen is 1:4. So 4 moles of hydrogen in the presence of enzyme nitrogenase by spending 16 molecules of ATP will release 2 moles of ammonia, and hydrogen gas will be released as a byproduct. So this is the equation of biological nitrogen fixation. Each time one bond of the hydrogen is broken down, and there what happens? Now see, like this. I'll just show you in first step. This is your nitrogen, right? And you have 4 molecules of hydrogen, 4 moles of hydrogen. So in the presence of the enzyme, one bond of the nitrogen will be broken down. Here one hydrogen will be attached, then one hydrogen over here it will be attached. What will be the intermediate product? NH double bond NH. Again, one hydrogen will come, one more bond will be broken down. What will be produced? Intermediate hydrazine, NH2 NH2, and then again one hydrogen will come to break this bond to release 2 molecules of ammonia. Along with this, 1 molecule of hydrogen is also released as a byproduct, and in doing this whole process, 16 ATP, please remember this number, 16 ATP is required. It is done in the absence of oxygen because the enzyme which is functioning for biological nitrogen fixation is sensitive to oxygen. Clear? So this much you have to remember for biological nitrogen fixation. Now you have converted your nitrogen into ammonia. Okay? But there is one problem, that ammonia is not the absorbable form. Roots cannot absorb nitrogen in the form of ammonia. So now next step is to convert ammonia into nitrate, and that step is known as nitrification. Correct? Chalo, then let's see what is nitrification. So nitrification is actually conversion of conversion of ammonia into oxides of nitrogen, that is nitrate. Remember, chemically nitrification, bachcho, it is oxidation reaction. It is an oxidation process which is done in 2 steps. So it is how many step process? 2 step. In first step, ammonia is oxidized to produce nitrite, as well as water is released, because from ammonia hydrogen will be released, which will combine with oxygen to form water as one of the byproduct, and the main product will be nitrite. Now this conversion, this nitrification conversion is done with the help of soil microbes like Nitrosomonas and Nitrococcus. Nitrosomonas and Nitrococcus are the 2 nitrifying bacteria which are chemoautotrophic also. These nitrifying bacteria, they are present in the soil. They keep on converting ammonia into nitrite, and this is actually an exothermic process. Lot amount of energy is being generated, and these bacteria, they use this energy to produce their own food. So they are chemoautotrophs. So this conversion is done with the help of bacteria like Nitrosomonas and Nitrococcus. Correct? These two are nitrifying bacteria. These are chemoautotrophs which use this energy to produce their own food. Right? So now you have obtained nitrite, but this is the first step. Second step of nitrification is further oxidation of nitrite into nitrate. No water will be released, but it is also an exothermic reaction. And this conversion is also done by another set of nitrifying bacteria found in the soil called Nitrobacter and Nitrocystis. Nitrobacter and Nitrocystis. These are also chemoautotrophic bacteria which allows the conversion of nitrite into nitrate. The energy which is released is then used by them to produce their own food. Correct? So now you have seen what is happening in nitrification. It is oxidation reaction with the help of chemoautotrophic bacteria. Now chemosynthetic bacteria, or you can say chemoautotrophic bacteria. Now you are having nitrate. What will be the next step? The next step will be uptake. So now your plants are going to absorb nitrogen in the form of nitrate present in the soil. Done. फिर क्या होगा? Plants are going to utilize them for protein synthesis. So now let's try to see that what happens inside the plant body once they take up the nitrogen in the form of nitrate. Again a trouble. Now that trouble says that plants, they cannot use nitrate to produce proteins. See, nitrate is the form of nitrogen which is preferred by roots to absorb but not preferred to produce protein. Hence now we have to convert this nitrate in the root cells back into ammonia. So that this ammonia can be utilized for protein synthesis. Ammonia was there in the soil, but what happened? Plant cannot absorb it. So we had to convert it into nitrate form. Now plant has absorbed, but now plant cannot utilize this nitrate to produce protein. So now what we have to do? We have to once again convert this nitrate back into ammonia in the root cells so that protein synthesis can take place. Clear? So now see what happens. So next step is conversion of nitrate back into ammonia. So that will be reduction reactions, and that is known as nitrate assimilation. So now next step is nitrate assimilation. Nitrate assimilation is actually reduction reactions. What is this? Reduction reaction where, these are reduction reactions. Why? Because here we are going to convert the absorbed nitrate back into ammonia so that plant can utilize it for protein synthesis. It is also done in 2 steps. It is also a 2-step mechanism. In first step, inside the plant cells, nitrate will be reduced to nitrite. First step. Correct? Now, how will this take place? The plant cells, they have their enzyme for the reduction of nitrate into nitrite, and that enzyme name is nitrate reductase. So with the help of the enzyme nitrate reductase, nitrate will be first reduced to nitrite. In the second step, this nitrite will further be reduced to ammonia with the help of another enzyme called nitrite reductase. Why? Because we are reducing nitrite. So the name of the enzyme is nitrite reductase. And in the first step, we were reducing nitrate. So the enzyme name was nitrate reductase. So basically in the next step we are assimilating nitrate back into ammonia so that this ammonia can be utilized for protein synthesis inside the plant body. Clear? So now you have obtained ammonia as well by the process of nitrate assimilation inside the plant body. Till nitrification, everything was happening in the soil. But after uptake, whatever is going to happen, it is going to happen inside the plant body. Ammonia is there. Now what plant cells are going to do? They are going to use this ammonia for their protein synthesis. Now let's see that how plants will prepare protein out of it. Yes, so that is called as fate of ammonia. So this is known as fate of ammonia. Fate of ammonia means let's see what is in the luck of ammonia inside the plant body, how it is going to be utilized for protein synthesis. So first step, ammonia is produced by nitrate assimilation. As ammonia is produced after nitrate assimilation, the first step that happens is protonation. Now, ammonia will be added, made to be added with one free proton to form ammonium. Now, ammonia in the form of ammonium is now ready to be utilized for protein synthesis. So first ammonia is produced, then the next step before protein synthesis is protonation. Protonation is done, ammonium is ready. Now we can use this ammonium to produce protein by two ways, as you can see on your board. The number one way is reductive amination and second is transamination. So first way is reductive amination and second is transamination. Okay. What is reductive amination? The name itself suggests to produce protein by reducing and transfer of amine group to a specific substrate. Reduction karwana, reductive means reduction, amination means addition of ammonium group. So ammonium group is ready. For reduction, reducing agent will be like your NADPH2, the nicotinamide adenine dinucleotide states, right? They will act as the reducing agent and you have a substrate that is alpha ketoglutarate acid. So now to this substrate, you're going to add ammonium group as well as you're going to reduce it in the presence of some specific enzyme to produce amino acid that is glutamate. Glutamic acid is one of your acidic amino acid which is produced by the reduction and amination of organic compound, a 5-carbon organic compound, alpha ketoglutarate acid. And this conversion is done in the presence of the enzyme that is glutamate dehydrogenase. Right, to see in reductive amination, what are we doing? In this process, ammonia, that is your ammonium ion, it reacts with the substrate alpha ketoglutarate acid to form glutamic acid along with reduction. And for this, the enzyme required is glutamate dehydrogenase. So this is my alpha ketoglutarate acid, the substrate. You're going to provide the ammonium which is ready to be utilized, plus the reducing agent is going to be utilized so that this substrate can be reduced. क्या बन जाएगा? Glutamic acid will be produced, which is one of the amino acid. Now you can use this amino acid for protein synthesis. This NADPH2 itself will get oxidized and water will be released as a byproduct. To do this, enzyme required is glutamate dehydrogenase. Right? So this is the equation of reductive amination. Moving ahead, the second method of protein synthesis by using ammonium is transamination. The second method of protein synthesis is simply transamination. Transamination means transfer of functional groups from one compound to another. Here we are having two functional groups which are going to be transferred, amine group and keto group. Let's read how. So what is transamination? It involves the transfer of amino group, NH2 group, from one amino acid to the keto acid. In return, that keto acid will pass on its keto group to amino acid. And in this exchange of functional group, the new amino acids for the production of protein will be produced. And this whole process takes place in the presence of the enzyme transaminase. So which enzyme is required for transamination? Your answer will be transaminase. Now let's see how transaminase is functioning and what is happening. So first I'll draw the general formula of keto acid and amino acid, and then I'll exchange their functional group. So this is the general formula of amino acid. You all know, you all have studied biomolecule. So this is the general formula of amino acid with R1 as a functional group, or R1 as a group which changes, then the nature of the amino acid also changes. Second is your keto group, keto acid with a keto group. So this is a keto group, and this acid with carb is known as keto acid. Now simply what you have to do, in the presence of the enzyme transaminase, this functional group, keto group, will be passed to amino acid. In return, this amine group from amino acid will be passed to keto acid. So there is simply exchange of functional group between two substrate. So yes, there is simply exchange of functional group in between two substrate to produce new amino acids. Now see, if you pass the functional group, what will happen? So R1, the carbon which is attached to R1 will now have keto group along with carboxylic group, plus R2 will have carbon attached to the amine group along with the carboxyl group. So this is your new amino acid. This one was your old amino acid, and now that is your new amino acid. Can you see the R group has changed towards the reaction, towards the reactant side? You were having, towards the reactant side you are having R1 group in the amino acid, but towards the product side, due to transamination, now you are having R2 group on the amino acid. So you can see that R group has been changed, and when once R group changes, what happens? The nature of amino acid also get changed. So for example, here it was X amino acid. So now on the reactant side, you have different Y amino acid. So with this also, this is also second method of producing new amino acids from the existing amino acids, right? And this whole process is done in the presence of the enzyme transaminase. I hope the fate of ammonia is also clear. Now protein is produced, amino acids are produced, which will then be used for your protein synthesis, and now the proteins will be utilized by the plant body for their building. It will be then taken up by the animals. A time will come that the organisms, living organisms, they will die. Then what will happen? Then comes the next step. Okay, there is no extra space. Let me add one page. Okay, wait. Yeah, so nice. So next step will be your Ammonification. Ammonification क्या है? Ammonification is simply the death and decaying of organic. Once the organic matter they die, the decay process start, and during the decay process, the ammonia gas is released, and this is known as ammonification. For ammonification, the bacteria involved are like Bacillus vulgaris. So Bacillus has some groups of bacteria which helps in Bacillus ramosus, Bacillus vulgaris, which helps in ammonification. Clear? And last step to complete the nitrogen cycle is denitrification. So here I am gonna write denitrification little bit. So last step of the cycle to complete the cycle is denitrification, where nitrate in the soil can be directly converted into nitrogen with the help of some soil microbes like Thiobacillus and strains of Pseudomonas. Clear? So this is how the nitrogen cycle is completed. My dear students, whatever we have studied for those bacteria which are performing nitrogen fixation in free living state in the soil. So like Azotobacter, Bergering, they are living free in the soil. So they simply perform nitrogen fixation like this. But what about those bacteria who are fixing nitrogen inside the plant body, like Rhizobium, like Frankia? So next we are going to study is about symbiotic nitrogen fixation. Okay, yes or no? Very good. Now we are going to start with the last topic of the chapter, which is symbiotic nitrogen fixation. I've already told you the nitrogen fixation which is performed by free living bacteria, but what about those which form symbiotic association like Rhizobium and Frankia? So you know there are two types of bacteria like Rhizobium, which gets associated with the roots of leguminous plants, and Frankia, which gets associated with the roots of non-leguminous plants. They are having the same type of enzyme, nitrogenase. They also require 16 ATP to produce 2 molecules of ammonia from 1 molecule of nitrogen. Everything that remains the same. The only difference is that whatever the free living bacteria they are doing in the soil, the same thing has to be done by these bacteria inside the plant body. That means if free living bacteria they are producing ammonia in the soil, so these bacteria are producing ammonia in the plant body. And in that case, if directly ammonia is produced inside the plant body, then there will be no such steps like nitrification and nitrate assimilation required. You know the reason why? Because in that case, there will be no uptake. Already inside the plant body, bacteria is there. All nitrogen fixation, all ammonia synthesis is taking place inside the plant body. So why plant root will take up the nitrogen from the soil when already they are having ammonia? So if there is no uptake step, then there is no nitrification. And if no nitrification is done, then there will be no nitrate assimilation. So symbiotic nitrogen fixation is better than those bacteria who are performing free living nitrogen fixation. Plants will prefer more of symbiotic. It is better because in that case there is no pressure on the roots of the plant to absorb nitrate, and for that there will be no nitrification step or further assimilation step required. Correct? So now let's see that how this symbiotic nitrogen fixation takes place. First, let's read it. Some types of symbiotic biological nitrogen fixing associations are known. Like for example, the most common is of legume bacteria relationship, that is Rhizobium with the roots of higher plants like lentils, pea, etc., clover, etc. Species of bacillus-shaped, rod-shaped bacteria, Rhizobium, has such relationship with the roots of several legumes such as alfalfa, sweet clove, sweet pea, lentils, etc. And one more example I have told you of Frankia, which gets associated with the roots of non-leguminous plants like Alnus and Casuarina. So here I am going to show you the steps of symbiotic nitrogen fixation one by one. Okay, so please stay focused. Suppose this is my root. This is the root hair. These are the epidermal cells of the root, followed by cortical cells and pericycle cell. Everything is there. But this is the root hair. The unicellular elongation from the epidermis of the cell. Root hair. Right? Now, root hair, this is the soil. These are soil particles. And in the soil particle are present your unicellular bacteria, rhizobium bacteria. So these are rhizobium. Now rhizobium, normally it is free living in the soil, but when it has to perform nitrogen fixation, so it has to enter inside the root of the higher plant. So now this rhizobium wants to perform nitrogen fixation. Even the plant also want. If it is the requirement of rhizobium, then it is also the requirement of bacteria. So what happens in that case? The root hair of the plant, they secrete a specific chemicals. They secrete specific chemicals to attract the bacteria rhizobium. So rhizobium, due to that chemical, it will be attracted towards the what? Towards the root hair, and they will get attached on the root hair. So now, due to the chemical released, the rhizobium bacteria will get associated, will get attached to the root hair. Once the bacteria, they attached on the root hair, now what they do? They curl the root hair inward. So next step, after the attachment of the bacteria on the root hair, it will induce the curling of root hair inward. Andar ki taraf the root hair will get curled. Once this happen, then your bacteria which were initially present on the surface of the root hair, they will start moving inside in a thread-like manner called infection thread. So infection thread will be formed. What you have to write? Root hair curls inward. So now the root hair is going to curl inward, letting all the bacteria present on the surface to enter inside the root hair, then forming an infection thread. When the bacteria are present in the infection thread, then their rate of multiplication is very, very high. These bacteria are dividing enormously to increase their number. This infection thread continues, crosses the epidermal cell and enters into the cortical cells as well as the pericycle cell of the root. Now infection thread, bacteria are actively dividing. They are just increasing their number tremendously, right? So these are the epidermal cells, followed by cortical cells, right? Then you know the anatomy, followed by endodermal, pericycle cells, all those, the anatomy. Now here is the infection thread, bacteria. Now these infection thread, then they enter into the cortical cells as well as some enter into the pericycle cell as well. So these are your cortical cells. Now once the infection thread reaches to the cortical cell, bacteria stops dividing and rather they start increasing their size, right? Apart from bacteria stop dividing and increasing their size, the cortical cells, they also start differentiating. They are parenchymatous cells. So they start converting themselves into hexagonal structures called nodules. So once the infection thread reaches to the cortical cell, bacteria stops dividing. They will start growing in size and some changes also occur in the cortical cells. They get converted into some hexagonal bodies called nodule. So next step is nodule formation, right? So now the cortical cells will convert them into hexagonal bodies called nodules having bigger sized bacteria. So these are rhizobium which have stopped dividing, rather they have just increased their size. Now for nitrogen fixation, anaerobic nitrogenase enzyme is already there with the bacteria, but you know nitrogenase will function only if anaerobic conditions are there. So for that, a special pink colored oxygen scavenger, a pink colored oxygen scavenger. Oxygen scavenger means the one which is responsible for removing oxygen from the cell. Leg stands for leguminous because the thing is happening inside the leguminous plant. So leguminous leg hemoglobin is produced both by the root cell as well as bacteria. So this pink colored chemical leg hemoglobin oxygen scavenger is a product of both plant cells as well as bacterial cell. So they together they produce this chemical leg hemoglobin which removes all the oxygen inside the nodule, creating anaerobic condition. And once the anaerobic conditions are created, the enzyme nitrogenase gets activated, and whatever nitrogen is there in the cortical cell, it will be utilized for the formation of ammonia. Correct? Yes or no? So this is how inside the plant cells, inside the cortical cells of the root, nitrogen fixation, ammonia formation is taking place. Now this ammonia is produced inside the plant body. It will be sent to different parts like leaves where they will be utilized for protein synthesis. Now no nitrification, no assimilation is required because ammonia is directly produced inside the plant. From the root given to leaves, then synthesis of protein by either transamination or reductive amination will take place. This is how whole nitrogen fixation functions. I hope symbiotic as well as free living is clear to everybody. There is no such difference except in symbiotic there is nodule formation, right? But in free living there is no such nodule formation. Simply taking inside the bacterial cell, ammonia is produced, nitrification, and then all those steps. So with this, finally we come to the end of the chapter, and as the rule says, as the policy says, we need to discuss some of the questions. The first question on the board is first stable product of biological nitrogen fixation. Here in the question, the type of nitrogen fixation is defined. So what is the first stable product in biological nitrogen fixation? Of course, it is ammonia. Hence, the answer of this question will become 1. I don't think anybody is having any doubt. Correct? Chalo, second question. Select the incorrect statement. So here you are having few statements. You have to tell which one is incorrect. The first statement says that macronutrients are required in excess of 10 millimoles per kg of dry matter. Correct? Macro means the elements which are required in huge amount. So first statement is correct. Second, they can cause toxicity if found in excess. Bachcho, macronutrients, they usually never become toxic because already every time they are required in the plant body in excess. Whatever amount you give to the plant body, they are happy. So usually in natural conditions, these elements, they never become toxic. How? They may become deficient, but toxicity is rare. So second statement becomes incorrect. Third, macronutrients are 9 in number. Absolutely correct. Macro are 9, micro are 8. So which option is correct? The incorrect statement is only B. Hope you have understood this question. Now moving ahead with the next. Enzyme for nitrogen fixation is. Options are given. You have to tell the correct answer. Is sensitive to oxygen? Yes, correct. The enzyme name is nitrogenase. Correct. Is it a molybdenum iron moiety? Correct. So all the three statements are correct with respect to the enzyme. Hence, the answer of this question will become 4, all of the above. Hope you have understood this chapter completely. Any doubts, any queries, you can post them whenever you feel. I'll surely reply to all your doubts, to all your queries. But it is very, very important that first you study. Studying is very, very important, and then comes the later part. Okay. So with this, your mineral nutrition is over. Next, I'll be coming up with the next chapter of plant physiology, that is photosynthesis in higher plants. Till then, stay tuned. Keep working hard. Keep smiling. Thank you, everyone. Bye-bye. Take care.